Welcome everyone to this next installment of the GHGA webinar. Today we're going to be talking about single-cell RNA-seq analysis, which is going to be presented by Florian. Before we get started, I just want to mention a couple of other webinars that are coming up. We have a talk about fair biomedical research data management coming up mid-February that we would like to invite you to. And then Florian is also going to give another seminar on Galaxy. You can register to both of these events via the website, ghgad.events, or use those QR codes. And now I want to briefly introduce our speaker of today. Florian has a bachelor in bioinformatics from the TH Wildau, a master's in bioinformatics from the Freie Universität in Berlin, and a PhD in bioinformatics from the University of Freiburg, in which time he was also in outreach and customer support, as well as a bioinformatic developer for Galaxy Europe, which again is something he's going to be talking to us about in one of the next seminars he's giving. And he has been a bioinformatician and scientific workflow engineer for GHGA since April 2022. And with that, I think I'm going to hand over to our speaker.
Yeah, exactly. So I want to tell you, well, basically some basics about single-cell RNA-seq analysis and the data. And I stress out basics because single-cell is a very fast evolving field with many technologies still also coming up, also with new tools and procedures. And you can fill not only a lecture, but a whole lecture series with this topic. So I will scratch basically the surface here of single cell. And I definitely recommend that the things I will talk about, that you read up about it again to get an in-depth knowledge about all of the topics that I mentioned here. I structured this presentation in 3 big parts. The first part is what is single-cell sequencing. So with that, I want to cover basically the laboratory part. And the second bigger part is how can I use sequencing, single-cell sequencing data. So what do I get from the laboratory? And so what, how can I use this data to answer my biological questions? And the last bigger part is what tools can I use? And with that, I go a bit into the direction what we are currently doing in GHGA with single-cell data. So these bigger parts have the following questions, which I want to answer for you. Again, the first part is laboratory. So there I want to answer what is the difference between bulk and single-cell sequencing, and how do I perform single-cell experiments? And quite often for most people, it's more interesting to know what kind of confounders do I have? So what should I look out for? And with that, we immediately go then into the bioinformatics, so the second part. So where I will answer again, what data do I get? How do I check my single-cell data, and how can I, or what can I do with that data? And the third part is basically, again, GHGA going into the direction of products. So we go into the direction of bioinformatics data analysis workflows. So how can I automatize this data analysis for single cell? And why is it important to implement workflows? And the last question is then kind of future direction. So what open challenges do we still have for workflows, but also in single cell data analysis? Between each of these parts, I will do a short break, so then you can ask questions directly, or so either Julia or Nicole provides, or not provided already, a link for Mentimeter, also a code, where you, during my presentation, you can already ask questions. So write into Mentimeter a question, and then I will answer it later after the webinar, or I will pick it up also during this Q&A. My slides, just briefly, that you understand how I structured my slides. First of all, they will be available, so you don't have to write things down. So you can look the things up again. Furthermore, you find in the right upper corner the symbol of the bigger section I'm currently in. So the symbols you have seen already. And on the lower left is kind of a take-home message for you for the slide, which is the most important message you should get from it. And in the right lower corner, you have also literature you can read up to. Again, I scratch here a bit of the surface, and the literature then helps you to read further. So I give you a direction, and then you can look into it in more depth with the literature. Okay. What is single-cell sequencing? So let's formulate the motivation behind it. I mean, I think all of you know kind of what bulk sequencing is. So you take, let's say, a tissue, and from this tissue, if you now stay with scRNAseq, you take a lot of cells which you lyse, and then you get a lot of RNAs from it. These RNAs you then sequence, and from these sequences you get your, well, whatever you're interested in, but most likely your gene expression. And from this gene expression, you try now to infer an answer for your biological question, which could be, for example, I have a healthy donor, and I have a donor which has a certain disease, let's say a cancer, specific cancer type. And I want to find now a marker gene which I can use to develop new therapies for this specific cancer. However, in order to find this marker gene, I have to know the or have to know which genes are generally different between healthy and non-healthy. And based on bulk, you get a lot of different cells, and therefore you get an average gene expression profile for your individual genes. And based on that, if you would investigate now a specific gene, it might happen that between healthy and non-healthy, you see actually nothing. So the gene stays in its expression the same. However, you know that biology is not as simple as just simply take a tissue, sequence, get gene expression profile. No, the tissue can be very heterogeneous. And that's the case for cancer, quite often for cancer. What that means is that your tissue consists of either several different cell types or several different cells, maybe from the cell type, on different cell stages. These are two possibilities. And based on this, each cell might have a different expression profile, which you can technically measure. And this we do in single-cell sequencing. So you take each individual cell, you lyse it, you get the RNA, you sequence the RNA, and based on that, you get an expression profile for each individual cell. And then later on, which I will talk about, you can then, which you can see here in the plot, each cell cluster and also visualize. And based on these clusters, you can assign, for example, different cell types. And now you might see, based on the cell types, this particular gene in healthy and non-healthy is different for a particular cell type, or even between different cell types, which gives you now better understanding about the mechanistics, about this tumor, and also the cell composition of this tumor. and therefore lead to possibly new target or therapy discoveries. So, however, there are also other possibilities you might use single cell to investigate. So you might be in general also interested about stem cell differentiation. So how stem cells develop to find basically a baseline for cell development. About tumor heterogeneity, I now already talked about. There's also the spatial. You can also nowadays do spatial single cell sequencing. So you get also spatial information about the cell type composition or cell stage composition during the development of a human, which is also important to know about different cell compositions in your body, and therefore also important for drug discoveries. Also, you can try to find rare cell types, which might be the cause of a disease, or single cell sequencing is, or single cell experiments are also generally very useful for drug testing. So if you already know you might have a good drug, then with single cells you can do a drug testing in a parallelized manner very efficiently. Okay, let's come to single cell experiments. So how do I perform now a single cell experiment? And you have nowadays two very prominent techniques. And one is a droplet-based technique. Most common or well-known is the 10x genomics. And they follow, or this technique follows or uses beads, where you have on the beads oligos, which captures your RNAs. They also have, which you can see here in the picture already, PCR primer, cell barcodes, and UMIs. I will talk about it in a few seconds, what they mean. And these beads then are combined in this technique with a cell. So you try to capture one cell at a time with these with these beads. And so in the first junction, you introduce to the beads your cells. And in the second junction, you introduce an oil, creating kind of an oil emulsion, where you capture your beads and your cells. And in this oil emulsion, you have now an environment where you then lyse your cell, and then your RNAs from your cell will be captured by the bead. You therefore introduce your primers, your barcodes, and in this mixture you have also all the reagents which are necessary then to perform the PCR amplification, as well as then the later on the reagents for the sequencing. So that's basically the droplet-based methods. Then you have well plate-based methods, and one example, or most prominent method, is the scRNAseq. And there are similar. The difference here is you don't use these beads and these droplets. What you do is you sort your cells with cell cytometry. One way is, for example, with fluorescent cell cytometry. So you can then also introduce fluorescent markers to your cells in order to already sort specific cell types you're interested in. And then each individual cell will be sorted into an individual well, where you then have again a mixture of primers and reagents in order to perform your PCR amplification and your sequencing. So these are the two most common techniques for single-cell sequencing. There are other ones as well, but these ones you hear very, very often. One important point, which you can here find also as a take-home measures, which is important for single-cell, which you also have, need to think about, is with bulk, you know that you can do biological replicates. In single cell, that's technically not possible because each individual cell is unique, because it's in a very every little time frame in a cell might change the transcriptional composition, and so each individual cell stands for an individual, let's say, sample. So in order to compensate for biological variants, you have to capture a lot of different cells and create a, I want to use the word cohort, but cohort used for patients. But you get the assemblement, a cell assemblement. You have to create a big cell assemblement in order to capture the biological variants. So to basically do what you do with bulk with replicates. Now let's come to the barcodes. So basically, how is the read library structured? And I have to add again, so this depends on the protocol, but you find quite often three things in your reads later on if you sequence your RNAs. And one, so which is untypical for bulk sequencing. So let's say, or a bit different to bulk sequencing. One is cell barcodes. Obviously, if you now have your RNAs and you sequence your RNAs from your different cells, at some point you need to figure out, okay, if I want to identify now different cell types or different cell, and later on do different cell, investigate cell clusters, I have to know from which cell this RNA comes from, and therefore you have your cell barcodes. One point I add here, which is important to know for if you read in the literature, quite often in the literature it is written that they don't speak about cells so much anymore, but about barcodes, which is a bit confusing if you come from the bulk sequencing area and you read about barcodes. Also in bulk, you know that you have different barcodes, and what in single cell papers, if you hear about barcodes, quite often what they mean is the cell barcodes. So what they are talking then quite often about the individual cells. A second molecular identifier, which you know from bulk already, is UMIs, unique molecular identifiers. These you also have to, or not have to, you also sometimes introduce in single cell, especially in the droplet, in the well plate. So for scRNAseq, you cannot really introduce UMIs. This has something to do because scRNAseq, in scRNAseq you sequence the whole read. In droplet, you only have the 3' and 5' end sequenced. And therefore, to not confuse you too much about it now, but in scRNAseq, therefore, you often don't see UMIs in there because of the full read sequencing. And in droplet, you find this, you can introduce UMIs. And you might know already that UMIs are introduced in order to reduce PCR amplifications. So PCR duplicate, PCR duplicates, sorry. So during PCR amplification, you introduce PCR duplicates. That means that for a read / transcript, you might, during PCR amplification, create not only one read, which you can see here in this example. So you have here a cell with 4 different transcripts, let's say, each differently coded now with an UMI, but later on in the sequencing, you end up with a read library. We have duplicates in it, meaning that some of these transcripts have more than one read. And these duplicates introduce a bias in your data, which you want to remove, obviously. And this UMI, so this barcode, so this nucleotide barcode, helps you in order to remove these PCR duplicates. So you might guess it already now. So you look for reads which have an identical UMI, and then you merge these reads together into one entity, so that you only count this particular transcript once in that example here. And a third barcode, which you might introduce, and you also know from bulk, is, I called it now, a donor multiplex barcode. Be careful now using that name. I introduced it now here to visualize it a bit better. So this means that you introduce a barcode for different patients. Obviously, you also want to know if you have a cohort. So with a cohort of patients where you have healthy, non-healthy, you will also want to know not only from where does my RNA come from cell, but also from a patient, from an individuum. And therefore you need, you can also introduce a barcode for that to identify this. Again, be careful with this donor. I named it now donor multiplex barcode, but obviously you can also use cell lines, and it's a cell line multiplex barcode. I think you get the idea. So this is about how I perform the experiments. How does my read library looks like? And now we come to some confounders, which you quite often see in single-cell experiments. In this graphic here, you see now an example. Let's imagine a droplet-based method. We have here our droplets capturing our cells, and the lines here are now our RNAs. And then one confounder which happen, and this happens very often, are doublets or multiplets. These droplet-based methods or well-placed methods are not errorless. And so what happens is that you might capture not only one cell, but multiple cells. So 2 cells or 3 cells in one of your environments here, and therefore, when you lyse these cells, you get a bunch of more RNA, which should actually not be the case, which is technically not the case for the biology behind it. And this RNA will be, because you have your beads in there, so you will give them a barcode, and later on you have higher read counts or gene counts, so reads for your genes or transcripts than you would actually have in truth, in the biological truth. This second confounder are empty, quite often called emptyplets, means empty droplets or wells. What happens? So the opposite to capturing more than one cell is you capture no cell. That does not mean immediately that you have actually no data. It can actually still mean that you get some reads from these empty wells. This can happen because in your cell mixture, so in your tissue sample, and then in your cell mixture, you have also cells which are in the stage of apoptosis, or they are have a lot of stress, so they are also degrading, or yeah, basically at the end you have in your cell mixture also RNAs floating around, which you then capture with these droplets, and therefore you might sequence reads which from cells which are actually not real cells, so they are dead cells or just floating around RNAs, and therefore you have in your data later on genes or transcripts with very low read counts, which you might want to filter out. What you in that connection also often see is that mitochondrial counts are then a bit higher if you're now looking into human samples, because the mitochondrium, so the RNA in the mitochondria is then also released, and therefore you capture this RNA as well. And a lot of mitochondrial, if you see a lot of mitochondrial RNA counts for this particular cell, this is quite often an indication that you deal here with an empty plate. Third problem is ambient RNA. And this is closely related to the second problem. Again, you have cells which are under stress, which are dying, releasing RNAs, which you have in your cell mixture. And if you now capture your cells, you might now capture your cells and this additional floating around RNA, so this ambient RNA, which you don't really want, because it leads again to overrepresented genes, because you have counts from genes, which maybe they are housekeeping. These are housekeeping genes, and they are actually counted more than it's actually what you get from your individual cell. And then you have spurious clusters. What this means, I will tell you later, and or higher cluster overlaps, or higher, what I mentioned right now, higher gene coverage. And the last problem are batch effects. These ones you also know from bulk sequencing. There, it is always a question if you should correct it for it or not. In single cell, it is a bigger issue. So we really have to take care of these batch effects. And with batch effects, this means you have your biological variants and your technical variants. And the technical variants is introduced by the person who is performing the experiment, or where the individuum, so where you took the cell material from which, so where the cell material came from which site. And these technical variants can be removed if you design your experiment well. Meaning, here on the left side is an example of a confounded design, where you take all the cells from individual mice here, and you plate them out, or you put them in each individual plate. So therefore, you create a really heavy batch bias. And in the right side, you see an example for a balanced design, where it's better actually to take the cells and now you scatter them. You can also do it randomly. Here it's a bit more organized, but basically you scatter these cells on different lanes, and therefore you get different technical variances, which you can later use to predict the technical variance and to remove it. So basically predicting the batch effects and to remove this batch effects from your data. What you quite often, so I will show you a picture. What might happen is that you see spurious clusters based on these batch effects. And when you do a batch removal, then your samples hopefully should mix very uniformly and not cluster individually.